neuronal activity represents the basis of information transmission in the nervous system. When there is a damage in the nervous system, such as in a neurodegenerative disease or after a spinal cord injury, the neural activity gets altered. And this alteration in the neural activity can have pathological consequences, such as neuropathic pain after spinal cord injury, or, for example, tremor in Parkinson's disease. In our group, we approach the neurological problems from the point of view of these alterations in pathological neuronal activity. The central theme of our research is the development and application of techniques for recording and processing of neural signals, with the general aim to decode complex neuronal activity. And specifically, we have 4 lines of research. The first is brain reorganization after spinal cord injury. The second line is the study of neuronal oscillations in neurodegenerative disease. The third line of research is to investigate the neural code particularly in the somatosensory system. And finally, the fourth line of research is the development of new novel neuromodulation techniques to treat neurological and neuropsychiatric disorders. We developed new analysis techniques to quantify the information transmitted by population of neurons. One of the most fascinating aspects of neuronal activity of large population of neurons is that when neurons interact, they tend to oscillate. And we build a solid base of knowledge about neuronal oscillations in neurodegenerative diseases such as Parkinson and epilepsy. And particularly in spinal cord injury, our results have practical implications about the needs of anesthesia and the control of anesthesia in patients, and some more far-reaching implications about the mechanism underlying long-term cortical organization after spinal cord injury and the consequent development of neuropathic pain. We are currently developing novel techniques for brain stimulation, for non-invasive brain stimulation, to prevent and or treat neuropathic pain in patients with spinal cord injury. And these same techniques have a great potential to treat also diseases of high impact for society, such as migraine or epilepsy. And in order to maximize the social impact of our research, we created a spin-off company with the Fundación del Hospital Nacional de Paraplegics, which is NEUREC. The closest collaborators in our own institutions are Dr. Juan Aguilar and Dr. Antonio Oliviero, who are both co-founders of NEUREC. We also have collaborators at national level, in particular Dr. Lisette Menéndez de la Prida at Instituto Cajal in Madrid. And we also have strong international collaborations with Dr. Alberto Priori at University of Milan in Italy, and with Karen Moxon at Drexel University in Philadelphia in the United States. And also I would like to make a considerations, because every time that we submit a paper for possible publication to a journal, that paper ends up in the hands of at least 2 additional anonymous investigators who act as reviewers for that paper. They provide their criticism, their suggestions, or the arguments for rejection of the paper. And we do the same with the paper of other investigations, so that we provide our criticism, our suggestions. And this process, which is all anonymous and doesn't involve any money, is called the peer reviewing process. So every scientist that publishes papers and participates actively to this peer reviewing process is technically collaborating continuously all the time with the entire scientific community.